Morning again, everyone, and thank you, John, for that great talk. And we'll continue another great tradition of another ASLO Award. And today we're giving out the Ruth Patrick Award. And can I have the slide? Oh, there we go. So thank you again to all the nominators, the letter writers, the committee, and the who've selected this year's award winner. So the Ruth Patrick Award honors scientists who have applied the aquatic sciences towards identification, analyses, and solving critical environmental problems. The 2025 Ruth Patrick Award is presented this year to Dr. Peter Leavitt for his broad scientific contributions for the field of limnology and his commitment to translating these findings for policymakers and for First Nations in Canada. Peter is a Fellow of the Royal Society of Canada, Distinguished Professor and founding co-director of the Institute of Environmental Change and Society at the University of Regina. He has dedicated his career to understanding and mitigating the impacts of human activities on freshwater systems, focusing on pressing challenges such as nitrogen pollution, cyanohabs, climate change, and the conservation of lakes. His research has gone on to influence legislative measures, including the Save the Lake Winnipeg Act and Saskatchewan environment requirements for the city of Regina. I'm jumping ahead to quote one of the letters, and I quote, "Peter's impressive and synthetic body of scientific contribution to understanding a wide variety of socially relevant problems in aquatic ecosystems. His work has an extraordinary impact on many sectors of society, including Indigenous communities in Canada who seek to regain sovereignty over their water." And I will end by again quoting, and the quotes are actually, the letters are amazing, so it's always nice to quote them. "Most of us endeavor to make an impact in one area of environmental science. Dr. Leavitt has delivered applied research outcomes and significant societal change in many areas, with contributions rivaling those of entire institutes." I have the pleasure of calling Peter up to the podium to receive your award.
Step into the light, Luke. I'd like to thank everybody who nominated me and the Academy. No, not the Academy. I also want to recognize that, a lot of the work that I've done over the last 20, 25 years has been in partnership with, rather than in service of, Indigenous people. And so I'd like to recognize that we're on the traditional lands of the people of the river, the Catawba Nation, as well as other nations. And also the work that I'm going to talk about is mainly in Treaties 4 and 6 in central Canada. And this is the homeland of 5 linguistic groups: the Nehiyawak, you know them as Cree, Saulteaux, Anishtabek, same thing, the Nakota, Lakota, Dakota, and then the homeland of the Métis. This is important for how this talk generates. Here's the problem. Water quality sucks. It's getting worse, and it is particularly bad in areas where there's a lot of human activity. Even lakes that are largely within the boreal area, such as the 10th largest lake in the world, Lake Winnipeg, are seeing significant degradation in our memory, in our lifetimes. In general, we know what's going on. Liebig's law of the minimum says that the productivity of a system will be regulated by the rate of supply of the most limiting element relative to demand. The corollary of that is, once that demand is met, it no longer limits. And this will be important. When we look at demand to supply ratios, we see that phosphorus often regulates the productivity of ecosystems, but nitrogen is not that far behind. And so typically when we look at surveys, whether they be lakes or maybe even coastal ecosystems, we see a pretty decent predictive relationship between the productivity of the system, the chlorophyll, how green it is, and the rate of supply of phosphorus. This is really driven home by classic, iconic experiments showing that the unique impacts of phosphorus are quite measurable, particularly in undisturbed boreal systems. I emphasize that because that's the past and this is the future. We're going to have 3 billion more people on the planet in a very short timeline. Looking at the median projections for the United Nations and the little yellow dot there, we're ahead of the predictions. So we're going to have more people. And the reason it's important is that the growth of the human population since 1960 has been supported largely by the supply of synthetic nitrogen fertilizers. The difference has nearly doubled the population on the planet. We call that the green revolution, the great acceleration, but what it is really an over-fertilization. It matters because going forward, we're going to need to nearly double the nitrogen application in order to make up the food for the people that are forthcoming. And we're going to do it in areas that have a long history of agricultural production. The reason this is important is initially a lot of farms were fertilized with phosphorus, particularly rock phosphate, and it led to this odd situation that there'd be an increase in the supply of phosphorus and eventually a saturation of the soils, which Steve Carpenter and others have modeled can take a millennium to dissipate. This onset of phosphorus supply, the saturation with phosphorus, leads to increased runoff and creates a condition that subsequent increases in nitrogen might change the water quality independent of phosphorus. And that's going to be the thesis of this talk. In these areas, we see expansions of cyanobacteria. You can pick your favorite type of lake, and they've basically all taken off since the mid-50s. And in surveys, both in Canada and the United States, we see that the single best predictor of cyanobacteria and the toxicity tends to be nitrogen. Again, kind of a warning sign. So I'm going to give you 3 little pieces here. First, I'm going to talk about the system that we've set up to look at some of these problems. Then I'm going to present the evidence for nitrogen effects. And then I'm going to talk about, more speculatively, why I think it's been beneficial for society. So this is where I work, an area of about 1 million square kilometers in the middle of nowhere. So oceanographers, I am sorry, we're a long way from the ocean. It's dry. There's not much marine air coming in. This is a grassland area. This is probably half of the projects that we worked on, the topics that we've worked on, because these are what's going on at the same time in this area. And you think, oh, throw up your hands, I can't deal with this. Well, the way you deal with it is you look for commonalities. And one of the key commonalities is how lakes are forced or affected by human activities. So we developed this conceptual paradigm. We call it the energy mass flux framework. Basically, it says lakes are open ecosystems. They're influenced, they're forced by the input of energy and matter. Energy is irradiance, heat from the atmosphere, kinetic energy from wind. Mass is water, solutes, suspended particles. They can arrive at the lake or in the catchment. So there's 12 different pathways by which our activities or natural variance is transmitted to lake ecosystems. So that helps you put all that stuff into perspective. The second thing that we've done is we've developed a long-term ecological research program, and I want to talk a little bit about that right now. It's called the CAPEL Long-Term Ecological Research Program. So we've taken a number of different approaches to study these systems. The one that would be most common to you and what you expect for an LTER program is a monitoring program where you contrast different lakes that have different properties and you try and extract the causal difference between the lakes from the differences in properties. I should say that for the next set of slides, the thing I want you to get out of it is in red at the top, the pictures that remind me what to say, and the people at the bottom are the ones who did all the work. So we monitor. And more recently, we've started with more intense instrumentation, high-frequency buoys. This is the work of Helen Balch at the University of Saskatchewan, as well as Carrie Finley. We've also done lake surveys. So we go out every 2 weeks to the core set of 7 lakes. But then in another 20 lakes, we go out seasonally every year. And then in another 100 lakes, we go out on a decadal scale. And the idea is that we can go from the in-my-lake mindset to scaling up to larger and larger physical processes. Because more data is better than fewer. We've also developed significant partnerships with different groups, Carrie with the Canadian Water Rangers Association and myself with the Indigenous populations. And basically I have very strong interactions with the File Hills Qu'Appelle Tribal Council, which is 11 bands in the southern part of the province, the Federation of Sovereign Indigenous Nations, which is 73 bands throughout the province. We also like to go back in time using paleoecology. What lives in the lake dies in the lake, it sinks to the bottom, and the law of superposition says that the deeper you go, the older it is, and you can quantify that. So you can go back in time here, from the old man to the less old man, and reconstruct what the lake says was important from its perspective. Like most aquatic scientists, we do a bunch of bottle assays, and we do sort of slightly larger scaled experiments, but we also do whole lake experiments, be it looking at the effects of winter droughts, testing alternative stable state theory by actually deepening a lake, or, as I'll talk about a little bit here, eutrophying lakes with nitrogen to see what the effect is. More recently, we've started to add a more remote sensing component, because at the end of the day, we can't be at all the lakes all the time, and so anything we can do to provide more information about the spatial and temporal variability in the systems, I think will help us understand what regulates ecosystem structure and function. Most everything says that the water quality sucks in the area that I work in. The pH is high, they're alkaline lakes, usually more than 1 milligram nitrogen per liter, often more than 150 micrograms per liter of phosphorus. Chlorophyll's typically over 50 micrograms a liter, and a lot of cyanobacteria. So the question is, how much of this is because of nitrogen supply to what is otherwise a very phosphorus-rich system? So I'm going to go through the evidence on different scales for the nitrogen effects. The first and most obvious one is who lives there. So this is a few decades of data from the least productive lake on the top and the most productive lake on the bottom. I've annoyed the phycologists in the room by taking off all the labels and just pointing out that the black areas are the areas for nitrogen-fixing cyanobacteria. Now, to fix nitrogen, cells have to undergo a fundamental cellular metamorphosis. So you don't do this unless you really need nitrogen. So there's a suggestion here that nitrogen is fairly important in several of these systems. So this nitrogen fixation is interesting because from a management perspective, people go, oh, well, you know, if nitrogen is being fixed from the atmosphere and there's unlimited nitrogen in the atmosphere, then there's no point in regulating nitrogen pollution. That's not true. What we did here is we estimated the total productivity of the system, carbon fixation. We multiplied it by the C to N ratio of the cells at that time. We estimated how much nitrogen must be needed to get that many algae. We then did whole ecosystem nitrogen fixation estimates with naturally occurring abundance mass budgets for a stable isotope. And while it's true that in some instances, I was going to point at the little screen down here, you won't see that. In some instances, there are lakes and years in which all of the nitrogen can be supplied by the atmosphere. The median is 3%, statistically insignificantly different from 0. In other words, nitrogen fixation, though it's at rates that are as high as anywhere on the planet, is just not enough to sustain the production that we see, suggesting that the input of nitrogen could degrade this ecosystem further. When you take those nitrogen fixation estimates and you throw them into a mass balance, you see two fundamentally important things. The first is that most of the nitrogen in the system is coming from two sources: inflow, these are riverine systems, but also release from the sediments. That's a key point. The second thing is, if you do a whole ecosystem mass balance, and that's a little cartoon on the right there, you find that the lakes capture more nitrogen than they release, and release more phosphorus than they capture. If you think about that, that means that the algae will not pick up things that they don't need, and if they do pick up things, it'll sink to the bottom and be captured by the lake. That means these ecosystems are fundamentally nitrogen limited. So, consistent with that, we've done your usual bottle bioassays, and if you add nitrogen or nitrogen plus phosphorus, we have a tendency to stimulate production later in the year. It can be suppressed early in the year. And you think, okay, well, this is just a little bottle, who cares? We'll do it 1,700 times over 30 years, and you get to see patterns that are a little more interesting, such as coherent changes in the rates of nitrogen stimulation of primary production in shallow lakes in this particular case. Nobody necessarily wants to scale to an ecosystem from a bottle, so we do larger scale experiments with different forms of nitrogen in lakes that we know are phosphorus sufficient and say, well, what happens? Here's a good picture. In the upper left, there's the reference system. In the upper right, we've added nitrogen. In the lower right, we've added nitrogen and phosphorus. And when we've added phosphorus alone, it clears the ecosystem. We think that's high grading the DOC and allowing the microbes to degrade it a little bit more fully, but the point is the blooms are driven by the input of nitrogen. Data shows pretty much the same thing. Not only are there more cyanobacteria, but the cyanobacteria are intrinsically more toxic. Their cellular quota is higher. Okay, let's scale up even further. This is a prairie pothole region. There's a lot of water bodies. Let's mess with a couple of them. So here's a whole ecosystem fertilization experiment that we're sort of halfway finished processing, where we've added just urea. Now, urea is the single largest nitrogen fertilizer on the planet, accounting for some 60% of all nitrogen use in farms. It's non-explosive, it's got a lot of benefits, and when you put it in these ecosystems, they get really green. So the maximum chlorophyll there is 500 micrograms a liter. That's a half a millimolar for people that prefer that. Reference system, no changes. We can go even further scale. The city of Regina drains into downstream lakes, and we can look at how changes in management in the city have affected the downstream systems. So here we actually calculated the amount of nitrogen and phosphorus released by the city for 100 years and took advantage of the fact that in the 1970s there was so much concern of phosphorus that the tertiary water treatment was added and the phosphorus was dropped by 85%, almost to baseline levels. If phosphorus controls primary production, then productivity should decrease. If nitrogen controls it, nitrogen has continued to increase, we should see that relationship. And when we look using the sedimentary record, we see that the abundance of cyanobacteria is a linear function of the amount of nitrogen released from the city over the period of time. In fact, there's a disproportionate response of the cyanobacteria as indicated on the right. So this is really saying this system is nitrogen limited. As a result, and I'll pretend like the city and the government regionally just said, oh yes, this is very convincing. It wasn't like that. But as a result of this work and other work, the city decided to upgrade their wastewater treatment plant to remove nitrogen. They went to a biological nutrient removal, which took out exactly the same amount of phosphorus, but in addition took out 95% of the nitrogen. What happened? Well, in the streams receiving it, and these streams are again nitrogen limited, we found that the reduction, the elimination of nitrogen from a nitrogen limited system downregulated production. We had fewer algae and we had fewer cyanobacteria. I'm not going to present the data. The benthos were pretty much the same thing. So if you take out a limiting element, it will downregulate production. The lake's responses have been slower, as you might expect. Rivers don't have much internal storage, lakes do. And so we've seen when we look at the total cyanobacteria, and remember the lakes are draining from west to east here with the two side channels. The ones on the right there that have the vertical dashed lines are the ones that are affected by the wastewater upgrade. There is a tendency to see that the cyanobacteria were a little bit higher after the diversion, but the reality is it actually proceeded by a couple of years. And when you look at the reference systems, there's no difference at all. So we didn't get that big burst in cyanobacteria that people assume you'll get if you have a lower nitrogen to phosphorus ratio. If you look at the microcystin concentrations, they're lower. Now, it looks like they're lower exactly when the nutrient treatment was occurred, but if you look at the reference system, it's more of a regional effect. So the lakes did not get more toxic as a result of removing nitrogen. What did happen is we got an increase in surface blooms, but again, it was not necessarily related to the changes in nitrogen to phosphorus ratio. What this is a ratio of chlorophyll in the surface to depth integrated chlorophyll. It takes a value of 1 when the water circulates well and there's no surface scums, and it gets higher and higher as you get a higher proportion of the biomass in the surface. We definitely see in the downstream lakes an increase in surface blooms, but if you look at the timing, it precedes the actual nutrient manipulation by almost a decade, showing that there's a climate effect, or I will say that it's a climate effect. So let's finish off with the benefits to society, and this is tricky to do. Like how can I prove this? Well, we've done nearly 450 independent, unique interviews on the research. The vast majority of these were local because that's where people care about the water. And I think that this is John Downing mentioned this morning, that if you get things into the media, people who are not necessarily interested in science will pay attention and become more engaged. The second thing is we can point to things that have affected the governance of water. The wastewater upgrade, that was nearly $250 million. In the upper right there, that's the legislative transcript for the Save Lake Winnipeg Act, which required the city of Winnipeg to spend $1.5 billion, or $1.35 American, $1.5 billion. And in yellow, though it may not be legible, it says, as a result of the research that we did. That felt pretty good. On the bottom were 2 projects that we worked with First Nations. One was to shut down a mine that was going to take twice the base flow of the river that supplied water to all of these lakes. Not sustainable. There was no cumulative impacts assessment. So the science came in with the Indigenous rights, and we took it to the federal court and won. On the right, there was a proposal by the current government to take hypersaline water and put it into a national wildlife area, transferring 75,000 metric tons of salt into the North America's oldest area. Finally, I think the evidence is with people, and this has been one of the most satisfying parts of working in the Northern Great Plains, is how engaged Indigenous populations are with the environment. Whether it's an educational TP, participating in powwow, talking with people, even sitting on parade floats, it's been a really impactful activity. So to sum up, this is kind of the way I think of things. You've got this great database that you play with, and you've got all your science, and you can use it in different ways. You can use it to address the environmental challenges. You can just pick out one that you like. You can get involved in the political side of things and look at the governance actions. Or you can also work with people, whether it's learning about ceremony, empowering youth through internships, putting on conferences for Indigenous science, working pro bono, or just supporting Indigenous rights. So in conclusion, I think I learned 3 things. You probably know all of them already. First one is that everything is connected. As ecosystem scientists, that's sort of baked right into us, but I'm not meaning just the environment. I mean people. I mean people in the environment. I mean economy and society, all of it. Second, from a paleoecological perspective, you'd understand, to understand where we are and how we got here, you need to look at the past. And to forecast the future, you need to look at the time series. Then the final thing is, this is a phrase from my Cree friends, we're all treaty people, whether that's an intercontinental trade agreement or it's an agreement between natives and colonists. We are all part of it. If you're here, you're in it, and if you're in it, you need to participate. So I'll just conclude by saying that, pick your favorite conclusion there, but I think the bottom line is quite simple. Science is for society, and we need to participate. Thank you.